and a bit nervous. But I'm really happy to be here to present some of the work that I've been doing in Dr. Philip Zerbe's lab on root diterpenoids in a very important bioenergy crop called switchgrass. And so let me advance and get this. So since you all are phytochemists for the most part, I'm not going to need to convince you that plants generate lots and lots of different, very important specialized metabolites to interact with their environment. So I thought it'd be fun to plug this quote that I often start my talks with into ChatGPT. And if you see the image here, it's pretty inaccurate. Plant doesn't really exist, but it's lovely. Got a lovely cloud of chemicals around it. Demonstrating this chemical diversity. But one thing that's missing in this photo is the below ground chemicals. So what about the roots? And over the history of phytochemistry, I'd like to argue that we've largely ignored the roots. I think we're doing better now, but in the face of a changing climate and pressures to grow more food and develop resilient crops, I think that this is going to be the next big frontier, and root phytochemistry is going to be very important. So our lab studies root diterpenoids. We study diterpenoids in all over the plant, but my focus is in roots. And the most famous of all diterpenoids are the gibberellins. And they're ubiquitous phytohormones across all land plants. GAs are activated in response to light, temperature, nutrients, and stress, and function in almost every aspect of growth and development in all plants. GA biosynthesis is conserved across all of these plants. We have a precursor, geranyl pyrophosphate, or GGPP here. We have two diterpene synthases that cyclize and rearrange this linear molecule to form chlorine. And then we have chlorine oxidases that add oxygen at various positions to generate diverse GAs. And these oxidases are cytochrome P450s. And we know that this is conserved, at least as far back as the bryophytes. And we believe that this is the ancestral pathway to all diterpenoid metabolism, including specialized diterpenoids. And over evolutionary time, this pathway, there's been gene duplication events and neofunctionalization, specialization, and subfunctionalization to lead to highly diverse specialized diterpenoid pathways in all plants. And so this is just a schematic of sort of the specialized diterpenoid pathways. So again, we have the central precursor GGPP. Then we have diterpene synthases. We have class 2 diterpene synthases that tend to come first, and then class 1 diterpene synthases that work on the class 2 products. And you can see in this model how we have these bifurcations that generate diverse diterpenoid scaffolds. And then we have cytochrome P450s that come along and functionalize these scaffolds into even more functional bioactive diterpenoids. In maize, this system is pretty well studied, and these specialized diterpenoids are really important phytoalexins. So we have caralexins and z-alexins that are really important in fungal defense, caralexins that are also important in pest defense. We have evidence that caralexins and delabralexins confer resistance to drought. And then below ground, we have specialized diterpenoids, the delabralexins that interact with the microbiome and even confer some defense against below ground pathogens. And these diterpenoids are specific to maize, and we often find that these specialized diterpenoids are lineage specific and tend to function in these defensive roles. So the model or the organism that I study is switchgrass. So this is a really important bioenergy crop. You can see here that it's very large. It's a perennial prairie grass native to North America. And it's valued for its high biomass, for feedstock, bioenergy production. But it's also valued because it's got this wide geographical distribution shown here on this map across the eastern US, extending up into Canada and even down into Mexico. And this geographical distribution covers lots of different climates, leading to high genetic diversity in switchgrass and variable adaptive traits. And so we're interested in studying the chemical diversity of switchgrass as we hypothesize that this could be important for its ability to adapt to these diverse environments. So you mentioned that our hypothesis is that this root chemical diversity contributes to switchgrass's ability to thrive in these different environments. And the goals of my project are really to explore this diversity, elucidate the biosynthesis of these compounds in switchgrass, and then characterize their function. And to do this, I use two approaches, so sort of a metabolomics approach, and then also a gene discovery approach. And then I use those different approaches to kind of inform each other and keep learning. So early on, we discovered a large and diverse diterpene synthase network. So this is very similar to that model I showed you where we have the class 2 diterpene synthases, and then we have the class 1 diterpene synthases. We have the GA pathway here, so this conserved pathway, and then we have a variety of specialized scaffolds. And some of these scaffolds we had seen before in other species, but some of them were also unique to switchgrass. And this happens to be the largest diterpene synthase network that we know of in crops. Sorry, somebody just came in. And so when we in our lab, we study representatives of both that upland ecotype and lowland ecotype. So namely, we look at cave-in rock and Alamo. Those names aren't that important, but we subject those to stress. In this example, I'm showing you a drought experiment that we did, where you can see that the upland plants, when exposed to drought, are clearly struggling, while compared to the well-watered ones, and then the lowland variety exposed to drought really survives pretty well and looks very much similar to the well-watered plant. And if we look at the diterpene synthase gene expression for that experiment, we can see that for upland diterpene synthase genes, I'll orient you to this. This is just a heat map showing gene expression. On the y-axis, we have the diterpene synthase genes. On the x-axis, we have leaf, which is in green, and root tissue, which is in brown, these two columns, and then well-watered, drought, well-watered, drought. And then I've highlighted the GA diterpene synthases up here. And really all I want to show you with this figure is that a lot of the specialized diterpene synthases are highly expressed in the roots of these plants and are responsive to drought. So we have this more red over here in the droughted roots. And then if we look at the lowland plants, we have, again, the same sort of heat map. The GA, this is hierarchical clustering, so the GA diterpene syntheses are now sort of spread out. But for the most part, you can see that the specialized diterpene syntheses in the lowland plants are also higher expressed in the roots. But interestingly, they're higher expressed in the well-watered plants. And so we have this differential diterpene synthase expression across the different stress types, which also solidifies that switchgrass and these two ecotypes are really great for studying this diterpenoid diversity. So we were pretty happy to see that. So we did a lot of work early on, like I said, on understanding this diterpene synthase network, and have lots of diverse diterpenoid scaffolds. But what about functional diterpenoids? And so a lot of my work has been to identify and characterize cytochrome P450s involved in the functional modification of these scaffolds. And to do that, I do sort of three different smaller methods. So I look at correlation, right? So we know how these diterpene synthases are expressed in the different ecotypes and across the different stress treatments. So I can look for cytochrome P450s that are correlated and co-expressed with those. We can also look at colocalization. So here, this is the genome map of switchgrass, and I've highlighted, they're very small, but I've highlighted where the terpene synthases are. So they're restricted to a few chromosomes, and so we can keep that in mind and look for cytochrome P450s that might be co-localized. I will say that we haven't found any specific diterpene gene clusters in switchgrass yet, but we do have some sesquiterpene ones that I won't talk about today, but we are keeping that in mind when we're looking for gene candidates. And then finally, and I think most importantly, we also can think about phylogeny when looking for these genes. All of the characterized diterpenoid pathways that have P450s, the P450s fall into a single clan, so the CYP71 clan, and they're restricted to just a few subfamilies. And because of that, we can use this information to narrow down hundreds of candidates, maybe to tens of candidates, and make it more manageable. And so I'll go into three of the subfamilies that we're studying in switchgrass: the CYP701A, CYP99A, and CYP71Z. And this is a phylogeny built with our P450 candidates from switchgrass, which is PV and in black. The P450s that are important in diterpenoid metabolism in rice in blue, and then in maize in green. And then we have a few oddballs from Soteria and sorghum as well. And so first, the CYP701A subfamily, which includes these GA choline oxidases. So that was the first logical P450 to think about, is finding this GA pathway. And if you look at what's been done in maize and rice, we have a choline oxidase that is conserved to the GA pathway, and then we typically have a second CYP701A that functions in a specialized pathway. So for maize, it's the carlexins, and for rice, it's the mamalactones. And in switchgrass, we have two as well. And so we were able to characterize those using our heterologous expression platform in Nicotiana benthamiana, and noticed or realized that we do in fact have one of the CYP701As that function in GA biosynthesis. So that's the one all the way to the left here. And then we have another one that adds a hydroxyl group here at the 2 position and is presumably part of a more specialized pathway. And so it was interesting that we still have this two CYP701As, just like we saw in maize and rice. And we suspect that you'll probably also see this if you look in other important monocrop crops. And so we plan to do that as well. And then moving on to the CYP99A subfamily, these are also really important, a little bit less studied, but really important for rice, mammalactone, and oryzolexin biosynthesis. And then our lab was able to characterize one in Ceteria that presumably also functions in a specialized pathway. And switchgrass had 9 of these. So this took a lot of work, but we did characterize several of them. And it significantly expanded what we know of the diterpenoid chemical network in switchgrass. So all of these are new diterpenoids that exist in switchgrass. I have 3 that I haven't quite solved yet. They're either still giving me trouble with purification or I just haven't solved the NMR structures yet. But several of these we have, and they range from a hydroxyl group function to a ketone, and we even have an aldehyde function over here. And some of them, this CYP99A50A, can actually do 2 things to one scaffold. So this is a pretty interesting family. We're excited about doing some more some structure function work with the CYP99As. So stay tuned for that. And finally, I want to talk about the CYP71Z subfamily. So this is a pretty well-studied subfamily in both rice and maize, and important for specialized diterpenoid biosynthesis. We have 6 of those in switchgrass, and so we characterize those as well. And these were interesting for a few reasons. First, the CYP71Zs catalyzed the addition of this furano group here, which we know from lots of other specialized metabolic pathways that this is a highly bioactive functional group. But also the CYP71Zs bypass this class 1 step. So instead of decorating the class 1 scaffold, they acted on the class 2 product. And so, and I was going to show that. And so that was really interesting to us because this is very, very rare. I don't know of actually any other pathways that do this. And so that was exciting. And so now that we have this huge diterpene synthase cytochrome P450 network and all of this chemical diversity, we are interested in understanding like what this means for the actual metabolites that switchgrass makes. So we're able to see a lot of these in switchgrass tissue, but a lot of they're likely not the final bioactive products that switchgrass is utilizing to interact with its environment. And so the other approach that we use is sort of a metabolomics focused approach, where we will expose switchgrass plants to stress. I've listed several of the stress experiments that we've conducted on switchgrass. And then we extract the metabolites, and then we also do transcriptomics on these tissues. And so I'm showing you again some results from a drought experiment. This was done by Kira Titka a couple years ago, where they noticed or observed an accumulation of diterpenoid metabolites in the roots. So again, this is a heat map, but now I'm showing you metabolites on the y-axis. We're showing you, again, those 2 varieties, Cave and Rock and Alamo. So this Cave and Rock is the upland, Alamo is the lowland ecotype. And we have leaves on the left and roots on the right. And I've highlighted here these metabolites, diterpenoid metabolites that accumulated in the roots under drought stress. And then we purified those and solved the structure for those. And were pleasantly surprised that they also contained this furano group. And if you remember from earlier, the CYP71Zs catalyzed the addition of this furano group, bypassing that class 1 step. And so we were able to fit our panacolloids, those diterpenoids that accumulate in the roots under drought stress, into our pathway. And I'm going to zoom in to get rid of some of the craziness. But the only difference between panacolloid A and the CYP71Z product was a ketone here at the 2 position. And then the only difference between panacolloid C and panacolloid A was a reduction of a double bond and then another addition of a ketone. And so this has now informed our gene discovery even further to look for additional P450s and reductases that could be involved in the rest of this pathway. And so I've been working on trying to identify the genes and enzymes responsible for this pathway. And so to do that, remember I go to gene expression, looking at the upstream genes of this pathway and look for correlating other correlating genes. And so this data I'm showing you on the left panel is the lowland and right panel is upland. The stress treatment here is different. This is now a methyl jasmonate stress treatment. So the top 2 figures are treated, and then the bottom 2 are mock, just buffer treated. And then this is gene expression. I believe it's FPKM over time. So it's a time course on the x-axis. So we can see that the upstream genes peak sort of around 12 hours and then taper off. And so we can use this pattern to look for P450s and reductases that also have this pattern. So we can do Pearson's correlation analysis. This is just using the CPS1, so this first gene as a bait gene. And we were able to identify an additional terpene synthase that can also catalyze this reaction, but I won't really go into that today. And then our next closest hit was an STR, so we're very excited to see that. And then we have some other genes or other functions here as well. And this is just showing you the top 10. We have tons and tons and tons of candidates. And if any of you are familiar with pathway discovery, it's quite challenging, and I'm still working on this. And I don't have the enzymes for these missing steps quite yet. I think I have them, I just haven't put them in the right order. But stay tuned for that. We can also think about localization, like I mentioned before, and you can see that the upstream genes of this pathway are actually restricted to the first chromosome in each of the subgenomes. And so I'm also keeping that in consideration when looking for these gene candidates. Okay, so that's sort of where I'm at with figuring out this pathway. I still have like about 1.5 years left to get it done. I'm determined. But what about other diterpenoid metabolites, right? I told you we were doing treating with stress and then extracting these metabolites and looking for diterpenoid diversity. And so to do that, we use liquid chromatography and tandem mass spec to identify diterpenoid metabolites. And in doing this with the various stress experiments, I've been able to identify 22 unique diterpenoid masses. So these are determined diterpenoids by, we have a in-house hand-curated library of the diterpenoids that we've characterized. And then we also have I use in silico fragmentation tools to also predict diterpenoid. And those predicted diterpenoids range from scaffolds, so 0 oxygens, all the way to 4 oxygens. And I even have 2 glycosylated diterpenoids, which is pretty exciting. And so from mass spec, we can say that metabolites are diterpenoids, but we really can't confirm that or elucidate the structure from that. And so we must purify the metabolites and do NMR. And so I've been working on that as well. So we use column chromatography. We do a large silica column, and then we also use semi-prep HPLC chromatography. And I've been pretty successful at purifying the panacoloids, but I've already shown you their structure. So these have already been purified by a collaborator, but I started with these because they are very, very abundant in root tissue. And it was a great thing to start to see if I could actually purify some of these compounds. So those were successful. In addition, I've been able to purify several of the other predicted diterpenoids. So here are just 2 that have 4 oxygens. These are just total ion chromatograms from the LC. But we need a lot of tissue for that. So I've been growing hundreds and hundreds and hundreds of switchgrass plants and extracting metabolites because we'll need like milligram amounts for NMR. So stay tuned for that as well. Because I am using this as a learning opportunity as part of my PhD to purify, it's pretty slow. But I've been working also with a fantastic phytochemist, Dr. Xingxing Li, from the last lab at MSU, who is much better at purifying things than I am. He was actually responsible for the initial work with the panacoloids and has been assisting in purification and identification of some of these compounds. And I wanted to highlight 2 of the structures that he has solved. So the first is this panacoloid-like compound that has an extra hydroxyl group here, which is now added to my search for this pathway to look for more cytochrome P450s. And then the second is this beautiful glycosylated diterpenoid that is also expanded my search for glycosyl transferases, 2 glycosyl transferases. And so, yeah, so that is sort of why I made this figure of having the metabolite diversity inform gene discovery and gene discovery inform metabolite diversity. So what about what these things are doing in plants? So that's like the major question, right? And Xingxing also led this project where he took root extract from switchgrass and soaked these metabolite discs. And then he inoculated the center of these plates with different strains of rhizosphere fungi and observed how their growth. And so he was able to observe that for this particular fungi, both lowland and upland extracts restricted the growth of the fungal hyphae here. And so this was done with diterpenoid fraction. So this isn't pure metabolite. And so we're hoping to, in the future, repeat these assays and additional assays with these purified metabolites. And we have different microbiome isolates and fungal pathogens. And so in summary, what we know at this time is that switchgrass is highly diverse and can adapt to different climates and soil types. It has a large and diverse diterpenoid gene network. And we've shown that switchgrass accumulates unique diterpenoid metabolites in the roots when stressed. And I didn't show this data today, but we have preliminary data that these metabolites are exuded from the roots into the surrounding soil. And so we'll be looking into that a little bit more. And we predict that switchgrass root diterpenoids may be involved in rhizosphere biotic and abiotic interactions. And so over the next year and a half, while I finish my PhD, I will continue to purify and solve additional root diterpenoid structures. We will conduct more bioactivity assays, but with purified metabolites on the microbiome isolates, maybe some microbiome communities, and in-house fungal pathogen library. And then I'll continue the gene discovery and characterization efforts for the panicoloids and these new glycosylated diterpenoids as well. And so with that, I would like to thank you for your attention. I'd also like to thank our lab, my PI, Dr. Philip Zerbe, and our lab is actually bigger than this. These are only the folks that have directly worked on this project. So I'm showing you kind of a summary of what we've done, but this has been a lot of work, and I've had a really talented team of undergraduates and then graduate and postdoc collaborators on this project. So they're awesome and should be acknowledged. And then I'd also like to acknowledge our collaborators at Michigan State University, Dr. Xingxing Li and Dr. Rob Last's lab. They even hosted me 2 summers ago and helped train me on some of the mass spec work. Dr. Xingxing Li is a fabulous phytochemist, like I mentioned. I'd like to thank Dr. Kira Titka, who did a lot of the early work on this project. She's now a PI at the University of Groningen, I think I said it right, and is a personal and professional mentor to me. And then I also wanted to mention that our lab is working closely with Dr. Sue Ree's lab and Dr. Hiroshi Maeda's lab to develop and improve the plant metabolic network. So this is a great database where all of our gene characterization, enzyme characterization work will go into this database. And it should be a really great resource for all of us phytochemists to use in figuring out what enzyme functions are, and then also predictive, some predictive power in the future. So thank you so much, and I'll take any questions. And I will try to pull up the video and stuff again so I can see people.
Yeah, thank you very much. Wonderful presentation. And for questions, yeah, please feel free to put them in this chat or raise your hand and ask them directly. I could start off with one question in the chat. Happy New Year and great presentation, Gabby, from Larissa LaForest. I would love to hear your opinion on where the major bottlenecks for your work is. It really seems like even getting an idea on the diversity of metabolites that plants produce is a huge undertaking, let alone translating that back to the biological understanding.
Yeah, okay. I think I'll stop sharing too so I can see people and people can see me. I think the major bottleneck in my personal work is the purification of each of these compounds. So we need lots and lots of plant material. If we don't already have the pathway figured out, so if we have the pathway figured out, we can make them make the compounds enzymatically and solve enzyme products that way. But if we just have these diterpenoid metabolites in tissue, we have to, they're typically in pretty small amounts. And so we'll need lots and lots of plants, like hundreds and hundreds of plants, which is a lot, very labor intensive. And then there's a lot of different purification steps to get pure compound for NMR. And sometimes even after all that work, they just don't want to purify.
Yeah, thank you. Michael.
Sorry, Lars, did you say my name?
Yes, I did.
Okay, couldn't hear it. I'm sorry. It just like cut out randomly. So yeah, thank you very much for the presentation. It's great to see the work on the kind of the enzymes that are branching towards this furan biosynthesis. Very cool chemistry on the paniculoids. I was wondering if you could go back to that slide. I just had some questions on your thoughts and your hypothesis for like why you drew it the way that you have it drawn. And if you have other thoughts from your enzyme candidates about what way it could go.
This
Oh,
slide?
cool. Yeah, it's got for me, it's got some sort of weird gray. Oh, it just went away. Okay. So I noticed you had the paniculoid A as a precursor to paniculoid C. And presumably, I guess the hypothesis here is that you have some oxidase that comes in and converts this furan to a lactone. And I was wondering if you had any type of further evidence to support why you think it is kind of going this way, or why you kind of drew it this way. Because at least from what I remember, like say if I was to chemically synthesize furan, I might start from what I remember in the chemistry, heating up an acidic mixture of a dione, and then basically you have formation of furan. Um, and so I was curious whether from your enzyme candidates, whether you have some sort of like enzyme interaction, because perhaps one of the intermediates which forms the furan, which is most likely a dione or diol or something like that, could undergo an oxidative change, and then you actually end up forming a cyclic lactone. So I was wondering if you have evidence to say that it comes from paniculoid A, or is paniculoid A kind of a an end product and it's kind of a metabolic dead end and it can't go to paniculoid C. Do you have evidence to support that, or is this just a way you chose to present it?
We don't have a lot of evidence of that, and I would really like to talk to you about this further, because
Oh,
uh, you seem to be a better chemist than me. But the only evidence that we really do have is we have been trying to knock down sort of this early pathway here, using VIGs. So genetics are pretty tricky in switchgrass because it's an allopolyploid and an obligate outcrosser. But when we knock down this CPS1 and its and 2 and anything else, we have a few that actually this is functionally redundant here. We do see a reduction in both of these.
yeah. Yeah, no, I was actually proposing that whatever en if there was another enzyme that interacts and kind of what I would imagine is actually the central intermediate is somehow the intermediate that's acting while CYP71Z25 works. And that kind of like will direct it towards C or A, depending on the presence of that enzyme or the, I don't know, the lability of that intermediate, if it exists for any amount of time in our perception. But uh, I also appreciate that it can totally go to, you know, your way of oxidation, or, you know, just formation of an alcohol and then further oxidation towards the lactone. It's it's interesting. I guess it would be for me an interesting prospect because it's such a juxtaposition between how you'd chemically synthesize it in a way and how you might uh, biochemically synthesize it. So it'd be like a very different way of approach. So I was just curious about that aspect of it.
Yeah, I'd like to talk to you more about this later. So is it
Sure,
okay if I send you an email?
yeah, we can talk, we can screen share, we can draw some structures, and I can kind of like articulate what I mean a little bit clearer if it's not. So
Yeah, that would be great.
good,
Because yeah, I
absolutely.
think especially with the work that I've done so far on this, I'm not 100% convinced that this is the way that it goes because of my exhaustive work in gene discovery and just not really um, getting there. So
Well,
I'd
I
like to
mean,
talk.
at least to credit you, I'd say the approach of using nBenth, which I believe that's what you're using for kind of like
Mhm.
assessing whether these things work, maybe not in vitro assays, is probably the best approach because having both of them at the both enzymes expressed at the same time, you might really start to see the formation of both rather than one or and it might be like, yeah, if I'm talking about my mechanism, it might be that C forms always with the reaction mechanism of A, but this enzyme pushes that boundary towards C more or something like that. So that's kind of what I'm proposing. But yeah, let's talk. I would love to.
Yeah, let's please, because I want to think about this in a different way because I've been thinking about it this way this whole time.
Sure. Yeah, thank you.
Thank you.
Okay, we have two more questions from the chat. From Emily Carroll. Do you have any tips or tricks for purifying diterpenes? Anything that stands out?
Patience. I mean, so just to go a little bit more into the methods, everything that we do first with a giant manual silica column and a super, super tight gradient. So we like for example, from like 100% hexane to like 1 to 2% adding a more polar solvent. And so it can be in collecting hundreds and hundreds of fractions. So I would say that I don't know if the way we do it is the most efficient, but it's been working for us and it's a lot of work. So I don't know if that's a tip because maybe other people have better tips, but it's difficult. A lot of manual work. I don't know if that helps.
Thank
I
you.
guess one other tip is that I found that you can buy like for preparative HPLC, you can buy these like super expensive columns, and we've been pretty successful with just using analytical columns to separate some of these compounds. So that's one tip to save money.
Thank you. Then there's one other question in the chat. Do you have seen any novel diterpenes when characterizing these enzymes in Benti from Billy Wen?
So definitely. So I think if I go back to just all of the work I did on the 99s, all the different P450s, most of those are novel. So all of those structures that we solved, and maybe I'm misunderstanding the question, but all of them we've only found in switchgrass. There are other, the functions of the enzymes themselves aren't very novel, right? There's other P450s that can add a hydroxyl group at a 3 position, for example. But the diterpenoids themselves are helpful. Did that answer your question?
We will see. Billy, please let us know if that answers the question. In the meantime, Nick, please go ahead with your question.
This is can you hear me all right?
Yes.
So this is actually more of a comment back to what Mike was asking about earlier. I am doing kind of parallel work, but in the Lamiaceae family, and I've got an enzyme that I had found and just published on recently, CYP76BK1, and it also makes a furan like your guys's. However, in going to this dione, we found it can also make the lactone rings that you guys are looking for. So, and it does based off our proposed method, it's sort of spot on the money where there's this intermediate dione that can cyclize kind of spontaneously is our prediction. We haven't done too much to really prove that, but an additional oxidation is sort of necessary with that intermediate to get a lactone that we have found anyway. And we see really trace quantities of something that looks like it could be these diones, but they seem to cyclize and with this carboxylic acid to make lactones, they cyclize pretty spontaneously. So we see small enough quantities where we can't really purify them that easily. But I think that they were onto something with that sort of comment on how they're cyclizing. It doesn't mean to say that there isn't a it couldn't get oxidized by some other P450 down the road, but it's quite possible you'll need to, co-express it with another gene if you can't see any lactone formation and just do a slightly larger infiltration with something. But I'm sure you're already doing that sort of work anyway. But it's just a comment more than anything.
Yeah, and you said you just published on that
Uh,
76BK?
yeah, I can link the DOI in the chat or something. You can check it out. Um, okay, but that's all.
Yeah.
Thank you. Yeah, the next one is Jörg, please.
Thanks, Lars. Hey, Gabrielle, that was a great talk. Fantastic work and very beautifully presented. Really enjoyed this. I have a very general question, and I also have a comment, but I go with the question first. This is just for my understanding. So I really like that you're going eventually into characterizing the functions of these diterpenoids in the rhizosphere context, and I think looking for effects on microbiome. So what are the possibilities of doing gene knockouts or expression knockouts in switchgrass?
So it's very difficult. So switchgrass, I think I mentioned earlier, but is a allopolyploid and an obligate outcrosser. There have been, I think, only 2 people to ever be able to do like a knockout in switchgrass. And our transformation facility has been working on a protocol for years. So the best we have right now is VIGS, so virus-induced gene silencing, which is pretty successful, but it is transient. So doing any sort of long-term studies, right, more than 2 weeks, is almost impossible with FIG. So even thinking about things like drought or changes in microbiome, I think would be very difficult.
No, but I think you will be coming up with good ideas for how to study these functions. And if you haven't had the opportunity yet, maybe discuss beyond discussion that you have in your own group and with your direct collaborators, discuss with Anne Osborne what suggestion that she might have. She published a very nice study, that was an Arabidopsis, of course, on triterpenoids and effect on microbiome changes in 2019 Sciences paper. So I think she might be a good person to discuss things with. My comment that I had is of an extremely general nature to your introduction when you mentioned that the root phytochemistry has been largely ignored, which is certainly true in the context of the type of work that you've been doing, where you go into discovering metabolic pathway systems, the genes, and the molecular biology. But the actual chemistry of roots in plants with regard to what was traditionally called natural products or secondary metabolites, there is a large amount of knowledge that goes back to traditional phytochemistry in the context of medicinal plants, because roots have always been a source for medicinal remedies and so on. So this is actually a source of information that I think we should all go back to and look into the old literature, what chemistry is known from root systems. And now with those beautiful tools that you've been applying, that is gene discovery using genomic approaches and metabolic approaches, learning more of the biology of root systems. But fantastic work. Thank you.
Thank you. I agree. Yeah, thank you.
Thanks. Yeah, there's one more question from Amanda.
Hi, Gabby. I wanted to ask, and we can talk about this later too. So I might have missed how many different switchgrass, maybe accessions or variety you've tested, but I'm wondering, since it will be challenging to do any kind of gene manipulation, if there's a way that you can exploit the knowledge you have on natural chemical diversity to then compare maybe different sessions that naturally produce different kinds of terpenes and accumulate different terpenes. And if that's something you will be able to do.
Yeah, I think that's something we're very interested in doing. So we only study the two varieties, one representative from each ecotype. There's only one genome, which is lowland. Actually, there's some genomic resources actually coming out for some of the other varieties. They're not as robust yet. And so we are very interested in investigating this natural diversity. So, growing all sorts of switchgrass varieties and profiling their diterpenoids. And then we could, yes, use those in maybe stress assays to see if these diterpenoids confer resistance to certain pathogens or drought, etc. So we're very interested in doing that. I don't know if I will get a chance to do that.
Yeah.
But maybe that's something for whoever takes this project on to the next steps.
Awesome. Thank you.
Thank you.
There's another question from Joe, please.
Hey, Gabby, great talk. Enjoyed it. I just had a question. You mentioned that the switchgrass was especially diverse in that diterpene network that you've been working out. I was just wondering if you could speculate a little bit more about why. Why do you think that plant in particular has such diterpene diversity? And then also a little bit about major minor compounds. Are some of those especially what the metabolic flux is going into? And it's a bunch of minor compounds? Just kind of a broad question.
So for your second question, we don't really know. A lot of the limitations for the intermediates are whether or not they ionize well on the mass spec. So knowing whether or not something's a major or minor compound kind of in those early stages with just one oxygen, for example, has been difficult. And then as far as, wait, your first question, I just lost.
Just speculating about why switchgrass so good at diterpenes.
Yeah, that's a good question. I think this like simple answer or whatever is just kind of what I already mentioned, which is that it's been able to colonize this, large and diverse landscape. But I don't know. We've been interested. It's got this pretty complex genome, and so we've done studies on, or like we've looked into like whether or not it's because of this complex genome. But then you look at more complex genomes like wheat or maize, and switchgrass still has like more that we know of right now. Switchgrass has more diverse diterpenoid network. So I don't really know. Yeah. Philip, do you want to add anything? Do you have any ideas?
I mean, as far as the geographical adaptation goes, no, we haven't studied this yet. But what we do see is that in addition to the wide geographical distribution of switchgrass all across northern to southern US and beyond that, we also see a high genetic diversity in ploidy and in general genomic variation. So that high ploidy, that high variation might certainly have been a factor in switchgrass maintaining and functionally diverging terpene pathways in general and the underlying enzymes. So I don't think that's the only factor. I would say that's probably a contributing factor to this. But what are the actual sort of adaptation drivers for this? I think that those are questions really I'm hoping to address when we dive deeper into understanding the biological functions.
Yeah, thank you.
Okay, I think we have one last question in the chat. What virus do you use for VIGs?
Yeah, so that's the foxtail mosaic virus. We have a methods paper from 2020. on that. If you just like Google switchgrass, or I can find it and drop it into the chat. Uh, 2022. It's only been 2 years. Oops, I sent that.
All right, thank you very much.
In there, Emily.
Yes, and if there's no more questions, I would like to thank everybody. This was a wonderful seminar, very nice discussion. Thank you all for this. And please stay tuned. We're sending some information if we will have a seminar in 2 weeks or not. Otherwise, we will see us again on the next iteration, which would be February 8th, I believe. Yeah, thanks again, and see you all soon.
Thank you.